So it's a pleasure to give a first contribution from the regulatory side to this meeting on the theme that supports the discussions in session 2 for this afternoon. I want to say some words about the role of the regulator, set up our framework for marketing authorizations, but also product labeling, summary of product characteristics, and patient information leaflets. And I think it's important to recognise those two separate responsibilities, the regulatory role not being all about the yes-no marketing authorisation, but also about the product literature. And to do that, I'll give a high-level description of our evidentiary requirements and some scientific and methodological principles and good standards, good practices. And then come back to this theme which a number of speakers have already picked up on, about promoting efficient collection and processing of data without challenging or compromising the different roles and responsibilities of the HTAs versus the regulators. So that being the framework, we are governed in the regulatory world by a series of legislative documents, and the text that defines the context of a marketing authorization is reproduced on this slide. Many of you will be well aware of this, and it sets up our key criteria around quality, safety, efficacy, and risk-benefit balance. So legislation provides that the marketing authorization may be refused if benefit-risk balance is not favorable, therapeutic efficacy is insufficiently substantiated, and then there are criteria around the quality of the medicinal product. The risk-benefit balance is defined as an evaluation of the positive therapeutic effects of the product in relation to any risk relating to quality, safety, or efficacy. And you'll note that the text under the ABC concentrates very much on the medicinal product in question, its therapeutic effects and its harms. It doesn't really extend, in fact you can say it doesn't extend at all, to relative comparisons with other medicinal products. And it does concentrate on this idea of therapeutic effects of the product and not so much where it fits into the overall healthcare system or into clinical practice. Decisions on benefit risk are made primarily through the CHMP, as I'm sure many of you know. Legislation also provides for the agency to give scientific advice, and as has already been mentioned today, scientific advice is given on a national level by national competent authorities, but also on a European level. And the legislation provides that the agency shall provide the member states and the institutions of the community with the best possible scientific advice, advising undertakings on the conduct of the various tests and trials necessary to demonstrate quality, safety, efficacy of medicinal products. And the framework for that within the European Medicines Agency is that the scientific advice is given by the CHMP along, as appropriate, with the Committee for Orphan Medicinal Products through the Scientific Advice Working Party. And the emphasis here is that this creates a really strong link between what we do in the meeting of the Scientific Advice Working Party and the scientific advice letters which are signed off by the CHMP, a really strong link between that planning phase, that scientific advice phase, and ultimately the assessment. So a really strong link to the CHMP, who are making the benefit risk decision and the scientific opinion on the marketing authorization. We go so far as to describe scientific advice, or at least I go so far as describing scientific advice to be morally binding for the CHMP. What does that mean? It's not legally binding, and there's a nice little caveat on the front of all the letters that says it's not legally binding. And it's not, let's say, scientifically binding, such that if the status quo changes fundamentally in the 5 years between the advice and the time of the marketing authorization, if something fundamental has changed, then all parties would have to recognize that. But in the absence of a fundamental scientific change, a real shift in the clinical paradigm, if you like, what we're doing in scientific advice is saying, look, if you complete these tests and trials and everything pans out the way we hope it does in terms of therapeutic efficacy for the drug and collecting a safety database from which we can make an assessment, then those tests and trials shouldn't be the source of any objection at the time of marketing authorization. And that tends to work. The assessors rely on the scientific advice. I'd argue that the other EMA committees also rely on the scientific advice. Scientific advice itself is rather well established now. Speakers already today have talked about doing this for 20 years or so, and numbers have gradually increased over that time, and probably are at an all-time high now, both on a European level and on a national level. It's also increased in scope from national consultations through to the European process, parallel advice with FDA, parallel advice with HTA, of course, the topic of discussion for today. And outside specific development programs, we can talk about broad scope scientific advice, or we can talk about qualification procedures for novel biomarkers or novel methodologies. And we can, of course, look at correlations between adherence to scientific advice and success at the time of marketing authorization with, I'm pleased to say, a positive correlation. What do regulators actually need to give a marketing authorization and to construct reliable product information? Well, aside from the pharmaceutical aspects and the quality development, there are some questions around basic science, understanding of the molecule, understanding of the medicinal product. These basic science questions go to support the development plan for the product, the development pathway, through the confirmatory development phase and into the post-authorization phase, but also, critically, to inform product labeling, the summary of product characteristics for the prescriber and the patient information leaflet for the patient. And without these basic science questions being answered in the early development program, constructing a robust and an informative product information is not possible. But then we move into the confirmatory studies where we try to establish therapeutic efficacy and through understanding the safety profile, we try to understand a favorable risk-benefit relationship. And as has already been reflected today, that then moves into the world of risk management plans and post-authorization studies. These points are highlighted on the text in bold as being more the topic for today's discussion of integrating the HTA needs in the development program. But that could be discussed. In terms of our standards, if you like, a high-level summary of our standards for establishing clinical efficacy and safety, we work pre-licensing almost exclusively with randomized controlled trials. And this is nice for us, of course. The methodology can be complicated, but it tends to give you a clean answer to a clean question. We work under the framework of good clinical practice. The database that's generated to support the evidence presented by the applicant is available, can be verified, can be interrogated. And we can check the robustness and the standards to which the trials have been conducted and the data have been collected. We have a number of global guidance documents, a whole series of International Conference on Harmonization, or ICH, guidelines across quality, safety, and efficacy. And these include, from my own sort of personal bias or personal interest, ICHE 1 about collecting safety data, ICHE 9 about methodological statistical principles for clinical trials, and ICHE 10 about choosing a control group in the clinical trial. And beneath the ICH, we have a large series of EMA CHMP guidelines looking at therapy area specific standards and methodological standards. Really? I don't think I can do it in two. Do my best. And actually, the next point comes to Mel's introduction, really, that we do constantly think in the regulatory world about balancing internal validity and external validity. The demand on us as regulators is to understand the properties of the intervention under study, the medicinal product under study in the relevant setting. Internal validity gives us good, clean answers to clean, precise, targeted questions. External validity gives us a more real-world answer so a different question. But at the increased risk of introducing bias and complications to the way the question's framed. So we could talk about the clinical trial population. A more homogeneous population gives us an answer which is more likely to be internally valid. a more heterogeneous population, an answer that's more likely to be externally valid. And you can see the tension, you can see the balance that we have to try and strike. Similarly with endpoints, we could talk about clinical outcomes, either short-term or long-term. We could talk about surrogates, we could talk about biomarkers, and different criteria, different standards would apply to establish therapeutic efficacy in different therapeutic areas. Another point just to pick up on this slide is the assessment of patient-reported outcomes or quality of life. And again, striking a balance between something that gives general information on quality of life and something that's much more targeted towards a particular disease area. And at the Scientific Advice Working Party, we spend quite a lot of time thinking about patient-reported outcomes that are very targeted towards a particular aspect of a patient's condition. And the parallel HTA advice that we've managed so far, the choice of control becomes a critical issue, is clearly a critical issue. We have a draft reflection paper in the regulatory system that talks about using a placebo control where it's ethical and feasible. Why do we do that? It's not so real world, is it? Well, we do that because we want to understand the absolute effects of the medicinal product under study. And if we go back to this slide looking at the standards, the risk-benefit balance being defined as the positive therapeutic effects of the medicinal product against the harms of the medicinal product. And of course, we need the context of the real world. But the placebo control gives us answers to questions on absolute effects for efficacy and safety and is highly valued in the regulatory world. Of course, a placebo control isn't always ethical and feasible. And in that case, we use a single active control, and then addition of an active control to a placebo-controlled trial is always welcome for context. Why can we use one active control when there are multiple drugs that are licensed, or if not licensed, are used across the whole of Europe? Well, again, we're not trying to establish relative efficacy. We're not trying to say drug X is better than Y. If the regulatory system was only to license the best available drug, we land ourselves in all sorts of contradictions, and we don't give the prescriber the option of an armamentarium from which to choose. So what we're using the active control to do is to say, here is a medicine that we understand. We understand its properties, we understand where it fits into the outside world. And if we make our comparison of the new medicinal product against that active control, we have some context for our decision. In terms of methodological approaches, the regulatory world really does strive to achieve unbiased comparisons and control of false positive error rates. And we're able to do that because we can force these direct head-to-head comparisons. We try to look at efficacy rather than effectiveness, and that's already been touched on today, focusing on the benefits that are observed in a clinical trial population attributable to the intervention under study. A couple of words on meta-analyses and modelling approaches. We're fortunate in that through the scientific advice process and through the scientific guidelines, we can describe the evidence base that we'd like to see for a risk-benefit decision. And the risk-benefit decision tends to focus on the trials that the applicant conducts, rather than other available external evidence. We don't usually have to rely on external evidence. We can make a decision based on internal evidence, let's call it, evidence that the applicant has generated. So when we use modelling approaches, it's primarily for interpolation within a data set, rather than extrapolation. And the obvious example that's been rehearsed today is from a 2-year trial to a 20-year follow-up. And to establish therapeutic efficacy, that's not usually needed. So people are already aware that we have post-authorization safety studies, registry database studies, meta-analyses, and work ongoing to integrate pre- and post-licensing safety data and risk-benefit decisions. Post-authorization efficacy studies are under discussion. But when to look for these efficiency gains that we want to talk about today? Perhaps it's too early to look in the time of exploratory development. That doesn't mean, by the way, that it's too early to talk, picking up on the earlier presentation. But maybe it's too early to try and integrate requirements during exploratory development. We could, of course, promote post-authorization studies that meet everybody's needs, but I think we've already heard today that that's probably too late. So we seem to be talking about building in information to meet the needs of HTA assessment into confirmatory trials. And we might think about different ways of doing that. We could either make phase 3 trials, confirmatory trials, have greater external validity, which is your point, or we could add additional assessments to provide information to meet the HTA needs. I think what we must absolutely guard ourselves against is some confusion in terms of roles and responsibilities and gold plating or using the highest common denominator, let's say, when we design trials. So we must not say, oh, the HTAs want a general quality of life outcome, so that should be our parameter by which we make our decision. Oh, and they need 10 years follow-up, so we need 10 years follow-up for the risk-benefit decision. We shouldn't gold plate, and we shouldn't use a highest common denominator type of approach. So after some words on timing, how then do we put everything into a single development track? And one question we can ask ourselves, perhaps slightly provocatively, but one question we can ask ourselves is, which HTA need are we trying to align ourselves with? And this speaks to the points made at the end of the last session, which is actually we can't necessarily say all HTAs have to use the same methodology, all HTAs have to make the same type of comparison, and therefore the regulatory system can align with that. We must concentrate on collecting the right data in an efficient way, collecting the right information to inform everyone's different needs. Certainly, if we go down the route of adding assessments to confirmatory development programs, we must be conscious of the increase in assessment burden for trial participants. Already, confirmatory trials are loaded with considerable numbers of assessments, scales, measurements, visits that trial participants and physicians have to undergo. But there are other initiatives underway which may help here in terms of targeted monitoring of clinical trials and in terms of, as we've already reflected, arguments around making the trials more pragmatic and increasing external validity. And then some final words on analysis and the data themselves, and thinking about whether we can have a single analysis track. I think a point for discussion in the sessions this afternoon would be around the format for data collection and storage, key data for how we monitor a trial in a targeted way, analysis plans which again don't try and answer the same question but try and use the same information in different ways, and data presentations and summaries that can meet the needs of different stakeholders. We could focus on some specific issues around handling missing data, for example, or the analysis of subgroups, which are very important in both the regulatory world and in the HTA world. And again, it's not about making the same decisions, but at least we could agree on some principles and some ways of approaching that problem. I think there is definitely, from our experience in the pilot program, an open mind towards the idea of changing and enhancing the confirmatory development program so it meets the needs also of the HTAs. And if that means some compromise on an unimportant parameter to what we would usually request in a regulatory guideline, then so be it. Harder to compromise is in the areas of bias, potential for false positive errors. The final word perhaps on guidelines, and there was a call for more guidelines to be developed, whether it's, you could think about methodological guidelines between the regulator and the HTA, more guidelines globally, EMA, FDA, and that's attractive in principle. But again, we risk this idea of gold plating or setting the highest hurdle. And the specific discussions we can have within the scientific advice about individual development programs is I think, really where we can show the flexibility that was called for. Much harder to do that in constructing a guideline which is applicable much more generally. But hopefully that gives an idea, an overview of the regulatory framework and some thoughts that can be picked up in the session this afternoon on our methodological standards. So thanks.
Excellent. Thank you very much, Rob.